This is a production of Cornell University.
Okay, my name is Zong-Yan and I'm from the Molecular Lab. Today, I'm going to introduce you GeneCAD, which is a plant genome annotation with a DNA foundation model. So we are going to share about what's the current landscape of the plant genome annotation and what GeneCAD used the conservation-driven architecture to do the genome annotation. And the third thing definitely is what the performance looks like. So before 2020, the N50 of the plant assembly is often shorter than 100 kilobase. And most of the things that we got is many contigs or fragmented genomes. And that is because most of the genomes are repeated polyploidies or there are heterozygosity cause this kind of barriers. However, with PacBio HiFi or Oxford nanopore technologies, the current N50 are commonly more than 10 or almost close to 25 million base pairs. And most of the assemblies can reach telomere to telomere per chromosomes. And for example, like in maize, wheat, barley, these kind of like highly repetitive proteins, plant genomes, we can almost reach like near complete genomes. And the other thing that's really impressive is the Earth Biogenome Project. So their goal is trying to sequence all the eukaryote species around the Earth. So their goal is try to sequence 1.67 million of the species. And as of 2024, there are more than 4,300 of the genome already been sequenced, which is more than 500 eukaryotic families. And as of this year, there are more than 2,200 of scientists across 88 countries in this project. So what's the current landscape of plant genomes annotation looks like is we can have the chromosome scale of the assemblies. Then let's look at what are the current tools or state-of-the-art tools for the genome annotation. So we can divide it into three different categories. So the first one is RNA and protein supported pipeline. And the most famous one might be Breaker or Osgotis. And the second one is the deep learning-based pipeline, and Helixer and Tiberius are in this group. And the third one is GeneCAD, and we are using a conservation-based pipeline. So let's talk about the first one, the RNA and protein-supported pipeline, and which is relative gold standard of our current genome annotation. So the input data will be required RNA-seq data and proteomic data. And they will train their model to do the HMM-based tool. It is relatively accurate, and it can also output alternative splicings or alternative transcript of each genes. So until now, lots of plant genome are still dependent on this kind of annotation tools. But the issue with this is if we're going to have a highly accurate annotation, it needs to be heavily depend on a good quality of the RNA-seq or proteomic data. And the other part is the deep learning-based pipeline. And compared to the previous one, the input data is only going to be the annotation file and the genome assembly. And it will train a CNN model or BioSTM, these kind of deep learning method, and also coupled with an HMM models. But the issue with this kind of pipelines is their junction boundary will relatively be blurred and typically require lots of training data set to achieve relatively high accuracy. And because of language model being recently popular, so we are using another way. It is called conservation-based pipeline. So we are use a DNA language model as the foundation. So we can get the embeddings of each genome or each transcript. Then we can start to train a BERT model or different kind of language model on different kind of models on top of it. Then we can do the prediction to get the GFF file. So let's go back to see what the current genome annotation looks like. So if we can have a relatively large and a high quality data, we can get a relatively good genome annotation. So let's look into what GeneCAD use conservation-driven architecture. So first, we definitely need to look into what are the language model that we are using. So the first one is developed by Dr. Jingjing Zhai in our lab called PlantCAD2. And it is a long context DNA language model for angiosperm. It is pre-trained on 65 different kind of flowering plant genomes with 8,192 base pair context window. And in this PlantCAD2, it is a single nucleotide resolution, so each base pair can get its own embeddings. And according to the models looks like, you can see it is a bidirectional and reverse complement equivalent. To understand this easier is if you input a DNA sequence into PlanCAD2, it can understand both forward strand and reverse strand at one time. And the second language model we are using, it is from a former PhD student in our lab called Amy. And in her paper, Real Gene, she pointed out that current pipeline routinely over-annotated. For example, in maize, even this is a relatively well-studied plant, around 28% of 1.8 million of the transcript models are non-functional, or it is a misannotation. And many of these genes are actually TE proteins or some pseudogenes that just looks like a gene. So how we are going to use real genes is there's a submodel called real protein. So they go through the core proteins across the pen genomes as the positive data, and the negative data will be the dispensable or the private gene. They also simulate some of the other false data like exon skipping or intron retention. Then the language model right now cannot use PlanCAD right now because it is a protein level. So we will run through ProTrans as a language model and get the embeddings, run the XGBoost model on top of it to do the protein evaluation. So we can see that large language model right now can help us learn the evolution sequence grammar. So let's look at what the current annotation cannot do for us right now. It is because the annotation sometimes disagree with each others. And according to Augustus, in their paper, they also claim that more than 70% of their protein being mispredict. And there are also some bias evidence. So RNA-seq or proteomic data, these kind of data are mostly focused on relatively few tissues and always focus on well-studied genes. So some of the genes will have never been studied. So how are we able to deal with this kind of issue? We introduced mask motif logistic regression. So this kind of method do not require any RNA-seq data or proteomics data, and we even don't need to fine-tune it. So we can generate a relatively unbiased sequence-only measure for the annotation quality. So how are we going to do the mask motif logistic regression? So let's give the translation initiation sites as an example. We notice that the start site is always start with A, T, and G. So in these three positions, we will give a special token like a mask token. And we can run through Planck-Caduceus-2 model to try to ask the model what's the probability of the model of the sequence to be A, T, and G. Then we will use a logistic regression to push the score even further. And we are not only run on the translation initiation site or the start site, we also run on the stop sites, donor sites, and acceptor sites of each transcript. So we can get a score across all different kind of genes and then get a score on each kind of transcript. So the next thing is we need to prove that it really works. So we use that proteomic evidence to confirm this score really works. And most of the time you can see that except Arabidopsis, many different kind of annotation are relatively low quality. So let's look into what Arabidopsis looks like. So you can see if it is a dark blue one, which means it is matched with the proteomic data. So you can see this dashed line is the mean of the proteomics transcript that is being matched. And if it is a red one, you can see that this is unmatched. But in Arabidopsis, because it is heavily studied and really deeply curated, so you can see all of their score are pretty high. However, if you look at some very well-studied crop, for example, corn, tomato, or rice, you can see if the score is pretty high, it definitely matched with the proteomic data, which is the dark blue one. But if it is not matched, you can see the NMLR score for these kind of transcript will be relatively low. So with GeneCAD, we can use the conservation-driven architecture to know that we can use the mask motif logistic regression to clean our training data to get a relatively good inputs training data for the model to learn. So let's talk about the GeneCAD architecture. What does it look like? So we need to start with the genome sequence. So just input the FASTA file, then we can get the PlanCAD2 embedding. So each base pair is then transformed into a context-aware embeddings. And that means it learns the millions of nucleotides across plant genomes. So with the embeddings, we next train a modern BERT. So it can learn like a central dogma. So it will output 3 different kind of predictions. So the first one is transcription, the second one is the splicing, and the third one is the translation. So if these 3 predictions match each other's, we can start to notice that, oh, this will be a gene. And we will definitely use an MLP head to really label it as a different kind of categories. And the other thing that I need to mention is we also use a CRF layer. So what does this going to do for us? So CRF layer is trying to really understand what biology looks like. So we will not allow a 3' UTR in front of the CDS and followed by the 5' UTR. It should always follow the biology rule, which is 5' UTR, CDS, and the 3' UTR. So if it's not match this kind of rule, we'll also remove that prediction. And finally, we will use what we mentioned previously, which is the real protein. So we will merge different kind of truncated proteins or just merge the small truncated protein into a longer one and run a real protein to do the quality filters. So this step can help us really remove the TE-like proteins or other junk open reading frame produced by GeneCAD. So using the conservation-driven architecture, we can notice that the structure decoding from the DNA embeddings can really help us to do the annotation pretty well. So let's look at the result. If you can see, it is the dark or the black one, which means in this category, for example, intergenic region, we can see that it's very confident. The model will predict this as intergenic regions. And as you can see, if we move to 5' UTR, this is also very similar to the biology boundaries. The UTR site there's a clear cut saying that this is the start of the 5' UTR. So you can see like a gradient color from light gray to black. However, if we see at the donor site, acceptor site, or the translation start site, translation stop site, these are cited as relatively conserved. So in GeneCAD, you can see the probability is always pretty sure that this is the CDS part. And also starts with this 3' UTR, you can see the gradient down back to the intergenic region. So the next one, we need to compare our tools with different kind of state-of-the-art tool. For example, what we mentioned previously, the breaker 3, the one we need to use the RNA-seq or proteomic data, or the one using the deep learning method is Helixer or Tiberius. So we will measure it in 3 different kind of way to do the evaluation. So the first one is nucleotide level. So we will compare each nucleotide one by one. So if we get the correct prediction, then we can label it as a correct. As you can see, GeneCAD is the dark blue one or dark gray one. And we are almost similar to the Helixer annotation, then both of us can get around F1 score to be 76%. But if we move a little bit more extreme, like exon level, this means the exon needs to be perfectly matched. If the shift even 1 base pair, the whole exon will be labeled as incorrect. So you can see a little bit stricter evaluations, GeneCAD start to win Helixer a little bit, and we can get the F1 score to be 70%. If we move it to the most extreme one, which is the transcript level evaluation, which means the 5' UTR, all the CDSs, and the 3' UTR, all this will be considered as a set. If any of them been missed, predict, even shift 1 base pair, the all transcript will be labeled as totally wrong. And you can see like the F1 score for GeneCAD can be like 44%. You can see relatively low, but the issue is some of the UTR in our annotation correct files that don't even contain the UTR site. And the other thing is GeneCAD can only predict 1 transcript per gene. And most of the annotation file, they will have multiple transcripts. So that is also the reason why you can see 44% as this number, but I think it's really good for the annotation. So let's visualize what it looks like. So if we look at the tobacco, because it can have diploid and allotetraploid, the first one is downloaded from Phytosome. So it's the first rule. And then the second one is the breaker 3. The third and the fourth one is the deep learning, Helixer and Tiberius. And the last one is GeneCAD. So let's look into it, what it looks like. So you can see compared to the first rule, which is the database one, you can see GeneCAD can predict relatively clear and without any other like false positive predictions. Even with all the tetraploid in tobacco, GeneCAD can also predict relatively clean prediction. So we won't really output some junk ORF or some TE to be labeled as a gene. So next, we are going to evaluate which part of the model is the most important structure for GeneCAD. So we start with the full GeneCAD and we do the ablation test. So we remove the post-processing data. You can see the F1 score jump a little bit. So these different kind of dots are different kind of species down here. And then if we remove the training data set from 5 genome down to 2 genome, and the accuracy also down a little bit. However, if we remove the CRF layer from this to this part, you can see this is relatively important parts because the F1 score decreased almost 5%. And definitely, if we remove the plain caduceus 2 embeddings, the prediction F1 score will be down to almost close to 0. So we can see which part of the plain CAD architecture, GeneCAD architecture is more important. So we can see that GeneCAD tool performance is relatively robust in a diverse plant, and then we can get a good result. So the take-home message for this talk is the current plant genome annotation is noisy and inconsistent. And with the large language models, it can contain the evolution grammars for the annotation. And the third one is mask motif logistic regression can help us provide an unbiased way to measure the annotation quality. And the fourth one and most important one is our GeneCAD can outperform the current state-of-the-art tools. So with that, I need to thank everyone in the Buckler's lab, definitely for all the authors and the people from OpenAthena and the professor from computational biology and the funding resources and the computing resources. And if you're interested in GeneCAD, you can scan the QR code to get the GitHub. And if you want to see more results, please scan the QR code to look into the bioRxiv. Thank you.
Open up for questions.
Hi.
Could you talk more about like the type of errors that the model makes the most, and like, do you see other avenues to sort of, get at those errors in the next version of this?
Yes, there are different kind of errors that GeneCAD produce, and how to go to the, okay, I can directly talk. So first is what I mentioned, the UTR site isn't that accurate. So we cannot really produce the correct UTR as the database. So the next step is we might need to include more different kind of sequencing method into GeneCAD to let the model to learn. And the other thing is we cannot do alternative splicing prediction. So we don't have alternative transcript. And this is also an issue, but it's a pretty important issue. Hi, Tim.
Zong-Yan. The slide where you had the figures with like the mismatch and match predictions for the 4 species.
You mean this one?
Further back, where you had like how the distribution of matches and mismatches. Further back.
Sorry. Which one?
This.
This one. Okay.
But can you describe what the score means on the x-axis?
Oh, yeah. Okay, I should talk about the slower. So Tim is asking what are the scores looks like. So we are trying to make sure the input data of our training data set is accurate. So we are trying to introduce MMLR score. So if the score is close to 1, which means it is a correct or it is relatively accurate or conserved gene. And if it's close to 0, which means this annotation might be wrong or it is not that supported by the plain CAD2 model. So we are able to get the score. If the score is relatively high, you can see that most of them are supported by the proteomic data, which is the dark blue one. And the score is relatively low, you can see it might not be supported by the proteomic data. So we want to use this score to prove that like we can clean up our training data set, not just input all the data set we download from the database to train the model.
This has been a production of Cornell University, on the web at cornell.edu.